Alcoholism is a complex disorder affected by genetic, epigenetic, and environmental factors. A picture of the genetic architecture underlying alcohol-related phenotypes is now possible because of genome-wide association studies. These tests can help you determine your heritability for becoming dependent on alcohol.
How prevalent is this trait in the human population?
In the U.S., around 14 million people are affected by alcohol dependency.
How many genes have genetic variations that impact your chances of becoming an alcoholic? Are these variations in specific genes that are associated with this trait?
Genetic factors account for about half of the risk for developing alcoholism, but identifying the genes and variations within them that cause this trait are oftentimes difficult. The strongest effects have been found for specific variants of genes that encode two enzymes involved in alcohol metabolism: alcohol dehydrogenase and aldehyde dehydrogenase. Accumulating evidence indicates that various variations in numerous other genes also have measurable effects. OPRM1 is an example of a gene that influences if you are likely to become an alcoholic. The gene changes the amino acids ASN to ASP. This SNP, known as RS1799971, can be the difference in a normal person and an alcoholic.
Can you explain the function of the protein or proteins coded by genes that are associated with alcoholism? Are these proteins made in specific cell types?
The normal function of the OPRM1 gene is to provide instructions for the creation of a protein called mu-opioid receptor. These receptors are part of the endogenous opioid system, which is the body's internal system for regulating pain, reward, and addictive patterns in an individual's behavior. The proteins produced by the OPRM1 gene
SNP chips
are
are
made
a
in the
type
nervous
of
system
marker-array
cells that regulate
DNA test
pain.
used to detect polymorphisms, or changes in the nucleotides of DNA, in a population. We are able to take the mutations we know are linked to certain diseases, then take a sample of your DNA and put it on a SNP chip and see if any of your DNA matches up with the
you be including in your
mutations.
SNP chip test, and why?
One SNP that is specifically prevalent in alcoholism is the RS1799971 SNP, where two genotypes are linked to alcoholism, AG and GG. The normal versions of this gene for someone who is not an alcoholic is AA. With this, carriers of the G allele tend to have stronger cravings of alcohol by 2.5 and 2.6 more times respectively. Alcoholism is about 50% attributable to genetics and 50% attributable to one's environment. This test, of course, cannot determine what your environment will be, and it is therefore very difficult to give the patient a specific percentage for their risk of developing
alcoholism was being at risk for genetic discrimination, but the Title II of the Genetic Information Act of 2008 prohibited genetic information discrimination in employment. This did not protect people with genetic mutations from insurance companies, but the known alleles that impact the trait may cause a slight increase in risk, whereas there might be other combinations of gene variations that can cause someone to be